what's the difference between Peptide Sameness Study and Biosimilar Comparability, and why does it matter? Here is a situation I see more often than you'd think. A pharmaceutical company is developing a synthetic peptide. They have done their analytical work, they are feeling confident, and then someone along the way, someone on the team says, should we be treating this like a biosimilar? And that one question, if answered incorrectly, can cause you months of development time and millions of dollars. Today I want to clear up that one and once for all. Hi, I am Raghuveer Singh Parmar, Director Scientific Affairs at ResolveMass Laboratories Canada. At ResolveMass, we partner with innovators to de-risk complex drug development through cutting-edge analytical strategies and deep expertise in challenging molecular modalities. So why does this confusion happen in the first place? Both pathways, peptide sameness and biosimilar comparability, are doing something that sounds similar on the surface. Comparing your product to a reference drug, you got a reference listed drug, your product, and the question is how close is close enough. But that phrase, how close is close enough, means something fundamentally different depending on which world you are in. And if you pick the wrong framework, you are not just doing extra work, you could be designing the wrong studies, generating data the FDA wasn't asking for, and worst case, submitting a dossier that doesn't meet the expectation of the pathway you choose. The downstream consequences are real, like delays, rejection, increased cost. So let's make sure you understand what separates these two approaches scientifically and also regulatory point of view as well. Let's start with peptide sameness, and I want to emphasize the word sameness here because it means exactly what it says. Peptide sameness study is used when you are developing a generic synthetic peptide, something like semaglutide, liraglutide, or similar molecules, and you are going down the ANDA route and the generic drug pathway. Your job is to demonstrate the exact molecular identity with the reference listed drug, the RLD, not similar, not comparable, identical. That means the same amino acid sequence verified to the later. The exact molecular weight confirmed by high resolution mass spectrometry, a comparable impurity profile. You are not just listing what's in your product, you are comparing it directly against the RLD, and the same physicochemical behavior, how it behaves under stress across the pH range, across various temperature. Now in practice, what does this analytical package actually work like? You are running high resolution mass spectrometry to nail the molecular weight. We are talking about sub ppm accuracy. You are doing LC-MS peptide mapping, which gives you sequence verification down to individual amino acids and also confirms disulfide bond connectivity, critical for many therapeutic peptides. You are running impurity profiling by comparing against R&D directly, and you are doing forced regression studies with orthogonal methods to understand how your molecule degrades. This is a tight, precise, analytical heavy exercise, and the reason it can be is because synthetic peptides are chemically reproducible. You control every step of synthesis, batch to batch. You can achieve true identity. That's the crucial point, and hold that thought for now. Now, biosimilar comparability is a different beast. Let's flip to biologics now, and this is where the science changes the whole conversation. Biosimilars are large complex molecules, monoclonal antibodies, fusion proteins, glycoproteins produced in living cell systems. And that word living is key. Living cells introduce variability, not because of poor process control, not because someone made a mistake, but because that's the biological reality. Glycosylation patterns shift, charge variants exist. You cannot make two identical batches of monoclonal antibody in the way you can make two identical batches of a synthetic peptide. So when the FDA says you don't need to prove identity for a biosimilar, that's not a lower bar. That's a scientifically honest acknowledgement of what's achievable. Instead, what you are proving is high similarity, and that requires a much broader evidence package. Structural and functional characterization, physicochemical, biological activity assays, PK/PD studies, pharmacokinetics and pharmacodynamics, immunogenicity assessments, because with biologics, immune response is a serious consideration. And often in clinical trials, to breach any residual uncertainty that analytical data alone can't close. The biosimilar pathway operates on what regulators call a totality of evidence framework. No single study is enough. You are building a weight of evidence case analytically, non-clinically, and clinically. The core distinction. Let me crystallize the core difference, because I think this is where the confusion really lives. The manufacturing origin is what drives everything. Chemically synthesized peptides, exact identity is achievable, so the regulatory expectation is prove it. Biologics from living cell systems, exact identity is not achievable, so the regulatory expectation shifts to prove high similarity with comprehensive data package. It's not that one pathway is easier and the other is harder. They are designed to reflect the scientific reality of what you are making. And here is a practical framing I like to use. If someone hands you a molecule and asks which pathway does this belong to, the first question isn't about the regulatory framework. First question is, how was this made? Is it a chemical synthesis? Peptide sameness. It's a living cells. It's biosimilar. Now, why does this matter from a scientific strategic standpoint? The peptide sameness pathway is generally the faster and less expensive. Not because the analytical work is easy, but because the scope is more defined. You are not running clinical trials, you are not doing multi-year PK/PD programs, your submission requirements are cleaner, your timeline is shorter. The biosimilar pathway, on the other side, is a multi-year, multi-domain commitment. You are building an analytical bridges, functional assays, non-clinical programs, and often a full clinical package. The investment is substantial, which is why early pathway determination is so critical. I have seen programs where the molecule was a synthetic peptide, no question, but the development team was approaching it with the biosimilar level scrutiny because they weren't sure, and that was the burning resources for no regulatory reason. Early alignment with your regulatory strategy, with your analytical team, with your clinical plan saves you from designing the wrong experiments for the wrong pathway. This is exactly the kind of challenge that comes to us at ResolveMass on almost regular basis. We work with companies developing complex synthetic peptides like GLP-1 analogs, cyclic peptides, disulfide-rich molecules, where the analytical characterization for sameness study needs to be all airtight. High resolution MS at sub ppm accuracy, full sequence confirmation by LC-MS, RLD reference impurity comparisons, forced regression with orthogonal methodology. But we also work for on the biological side, like molecules where the question isn't are you identical, but have you characterized your variability well enough to make a similarity argument. Those are very different analytical questions and they require very different experimental designs. What I have found is that the teams who come to us early before study design, not after, consistently navigate these pathways more smoothly, because getting the analytical strategy right before you generate the data is far less expensive than trying to fill analytical gaps retroactively. So let's bring this home with a few clear takeaways from all this session. I listed down. Number one is peptide sameness is about molecular identity, exact, proven, analytical. It belongs to the generic pathway and synthetic origin is what makes it possible. 2. Biosimilar comparability is about high similarity, not identity. It reflects the biological reality of large molecule manufacturing and it demands a much broader evidence package. Number 3, the regulatory expectations aren't arbitrary. They are rooted in science. The complexity of your molecule and how it's made determines what you can demonstrate and what regulators will ask for. Number 4 is choose your pathway early. This isn't a decision to revisit at submission. It shapes every study you design, every assay you run, every dollar you spend. If you are working on a complex peptide or biosimilar program right now and you are wrestling with how to frame your comparability or sameness strategy, I genuinely encourage you to think carefully about where on this spectrum your molecule sits. The science is the guide, the regulation follows the science, and if you want to talk through the analytical strategy for your specific molecule, what studies make sense and what the data package should look like, that's the conversation we are always happy to have at ResolveMass. Drop a question in the comments, reach out to us directly on our team and connect with us through the link below. Thanks for watching and I'll see you in the next one.